Welcome to my channel. In this lecture we will discuss about the prions. The characteristics of prion. The prions are the infectious protein particle that cause a neurological diseases. Prions are the infectious protein particles that cause a neurological diseases. They do not contain DNA or RNA, and so they cannot be digested by a nucleases. We know that the nucleases are the enzyme which cleaves the nucleic acids like DNA or RNA. So, as the prions are not any kind of a DNA or a RNA, so they cannot be digested by a nucleases. They are only a protein molecules, and this protein molecule is infectious in nature, and this causes a neurological diseases. As the prions are the proteins, they can be digested by a proteolytic enzymes. The proteolytic enzymes are the enzyme which cleaves or which digests proteins. So the prions can be digested by the proteolytic enzymes. Prions are resistant to the radiations. As I told, the UV light, gamma rays, X rays, these radiations kills the pathogen or kills the microorganism. But the prions are resistant to the such kind of radiations. Prions are resistant to the high temperature up to the 90 degrees Celsius. They can withstand for a high temperature. As said earlier, these are the proteinaceous infectious particles composed of a single protein, that is a PRP protein. All mammals contains a gene that codes for a primary sequence of amino acid in a PRP protein. The PRP protein is nothing but a prions, and all the mammals contains a gene for a coding of that PRP protein. The prions are having two stable tertiary structures. One is a normal functioning structure and other is a disease causing structure. The normal functioning structure, it is with a alpha helix and called as a cellular PRP. Normal functional structure of a PRP protein is with alpha helices and called as a cellular PRP. Whereas the another one, that is a disease causing form of a PRP protein, it is with a beta sit and called as a prion PRP. The prion PRP convert the cellular PRP. We know that the cellular PRP is a with a alpha helices, and a prion PRP is a disease causing form, and the cellular PRP is a normal functional alpha helix PRP. The prion PRP convert cellular PRP into a prion PRP by inducing some conformation changes. When these two forms of the PRP proteins comes together, then the disease causing form convert this normal functional form into a disease causing form. The basic structure, normal contains 200 to 250 amino acids. These amino acids are twisted into a one cord and forms a coil-like structure, that is a helices. The mutated and infectious form is built from the same amino acids, but a different shape because of the mutation. In the image, here is a normal prion and here is a mutated prion. The number of amino acids in both the prions are same, but their shape is a variable. About the size of prions, these prions are 100 times smaller than that of the smallest virus. The size of prion is very low or very less, 100 times smaller than that of the smallest virus. How the prions are different from bacteria and virus? We know that in a bacteria there is a presence of nucleic acid, proteins and other components, but in prions there is a presence of only a protein, not nucleic acid. So prions are not considered as a bacteria and these are not considered as a virus. They are extremely resistant to the heat and the chemicals. Prions are very difficult to decompose or degrade biologically, and hence they survive in a soil for many years. These are the proteins. The prions are proteins that are unique in their ability to reproduce on their own and become infectious. They can occur in two forms, PRP sen and PRP res, PRP-SEN and PRP-RES. These are the two forms of the prion proteins. Both PRP-sen and PRP-res are made up of the exact or same string of amino acids. We know that the amino acids are of the proteins. PRP-sen protein and PRP-res proteins are made up of a same string of amino acids. However, these two forms are of the different shapes. PRP-sen is produced by a normal healthy cell. The SEN stands for a sensitive because this version of a protein is sensitive to being broken down. So this protein get broken easily. It is sensitive for a breaking, so it is called as a sen or a sensitive. PRP-sen is present mainly in neurons in a brain and also found in other types of cells. The scientists don't know the exact function of a PRP-sen protein, but there are the evidences that the PRP-sen protein is involved in a communication between neurons, cell death, controlling sleep patterns. And interestingly, the mice that are genetically engineered to produce PRP-sen are looking healthy, and it was concluded that the PRP-sen protein form of a prion is non-disease forming one. The second type of protein, prion protein, is known as a PRP-res protein and it is disease causing form. The organism with it develop a spongiform disease. The RE stands for a resistant because this version of a PRP is resistant to being broken down. It does not get broken easily. So resistance form is a disease causing form. Unlike other infectious agents like bacteria, virus, rickettsia, protozoa, they contains genetic material. Unlike other infectious agents, prion do not contain genetic material. However, once they infect an individual, the prion can replicate. Now the question is that how the prion can replicate. They are a protein in nature, and how they can replicate. they are not any kind of nucleic acid or any kind of cell, how they can replicate or how this is possible. Scientists are still working out the such details, finding out the evidences. When the PRP same protein comes in contact with the PRP-RES, it is converted into a PRP-RES. The PRP-RES is a disease-causing version of a prion protein. Any sensitive prion protein comes in contact with the ration protein, it gets converted into a PRP-RES protein. This results in a chain reaction and that multiply a copy number of a infectious prion. Because of their abnormal shape, PRP race proteins tends to stick each other, and over time the PRP race molecules stack up to form a long chain, and this stacked long chain is called as amyloid fibers. Amyloid fiber, it is a long chain of a PRP race protein. The amyloid fibers are toxic to cell and ultimately kills the cell. The cells are called astrocyte crawl through a brain. These cells are digesting a dead neurons, leaving a holes where the neurons used to be, and amyloid fiber remains. Here in this image, here are the astrocyte, microglia, dendrocyte. The normal form of a protein is called as a PRPC, while the infectious prion is called as a PRPSC. The newly formed prion can then go to convert a more protein themselves. This triggers a chain reaction and produces a large amount of prion forms. Prions cause other similar protein to also misfold and lose their function and cause a disease. Then next, that is a prion diseases. Prions are responsible for a transmissible spongiform encephalopathy. It is a one kind of disease in a mammal. It includes a bovine spongiform encephalopathy, that is BSE. The bovine spongiform encephalopathy or pathy it is also known as a mad cow disease, and it is observed in a cattle, whereas same version, that is a CJD in a humans, Creutzfeldt-Jakob disease. All known prion diseases affect a structure of brain and other neural tissue, and hence an infection by a prion leads to a fatal, and such kind of infection are untreatable. The history of prion in 1730s, there was an outbreak of a crappie. The crappie is a disease of a sheep. The English sheep were gone infected by this disease. The causative agent for a crappie was one prion. In 1950, the high-level outbreak of a disease kuru were appeared in a New Guinea, people of New Guinea. In 1960, the scientists experimentally transmit a kuru and a CJD to a chimpanzee and concluded that a transmissible nature of the disease, it was a one protein. In 1980, 60 people died from a CJD after being infected by a contaminated surgical instrument. 85 people die after receiving prion impacted growth hormone injections. In 1982, a scientist Stanley Brushnell coined the term prions, proteinaceous infectious particle. The prion, the two letter PRI, that is for proteinaceous, IN for infectious particle. The scientist Stanley Brushnell coined the term prion. Highly purified PRP race protein is shown to be infectious, and for this discovery of a PRP race protein as an infectious prion, the scientist Stanley Brushnell got a Nobel Prize in Medicine in 1997. In 1985, scientists identified the PRP gene, and also they discovered a unimpacted people produces a normal form of PRP gene or a PRP protein. The CJD or Creutzfeldt-Jakob disease, the most prevalent of the pungiform disease, occurs spontaneously in 1 out of a million people. 10% cases get inherited because of a mutation in a PRPN gene. The people of age in range between 50 to 75 got diseased because of this CJD. Symptoms like dementia, muscle twitching, vision problems. Second disease, fatal familial insomnia. It occurs in humans. All cases are inherited because of a mutation in a PRP gene. People age in a range of 36 to 60. Disruption of sleep, wake cycle, coma, and even death also. Scrappy, it is a disease of a sheep and a goat, and there is no evidence of this scrappy get transmitted from a sheep to the humans. Bovine spongiform encephalopathy in cattle or a mad cow disease in a cattle. Chronic wasting disease in a deer. Kuru in a human. This is about the Creutzfeldt-Jakob disease. Because of infection of Creutzfeldt-Jakob prion, the brain get shrink down and deteriorate. A controversy is there. There is no any kind of DNA of RNA. The proteins are there, and how the protein get replicated. That's it. Thank you very much. If you like my video, subscribe my channel and share my videos.